All right. Well, thank you everyone for coming to this session, Saving Vision and Treating Ocular Disease. My name is Dr. Rishi Singh. I am the incoming chair of ophthalmology at Mass Pioneer and Mass General Brigham. And I'm joined today by this expert faculty panel. And my job is very easy. I just make them all look really smart, which they already are, so it makes it very easy. First, Dr. Petr Baranov. He's specialist in retinal regeneration and stem cell biology at Mass Pioneer. I have Dr. Reza Dana. He's the director of the cornea refractive service at Mass Pioneer as well. Dr. Erin Kimbrell. She's senior vice president, head of cell and gene therapy research at Astellas Pharma. And I have Nazli Zabrast, who's also assistant professor of ophthalmology at Harvard Medical School and medical director of the glaucoma imaging at Mass Pioneer. So thank you all for joining me. And this is an interactive session, so I do have an iPad here that you can ask questions from the app, and I'm happy to take them during the panel. And just kind of start off, maybe start with Nazli. You know, we look at obviously this tremendous innovation in ophthalmology. Many of us, even though many of us look younger, have been here in the field for 20 plus years, and the field has changed so much. So why don't you talk about the last 2 decades and one of maybe the most single transformative area in your innovation area that has occurred in the past 20 years?
Yeah, so I think my perspective may be shaped by the work I do as a data scientist and a clinician, but I think the shift that we're seeing now, moving away from this one-size-fits-all approach in medicine to more of a personalized medicine approach is what I think is really transforming how we care for our patients. In glaucoma, I think the advent of AI combined with genomics, big data analytics has really allowed us to understand disease phenotypes and trajectories better, and combined with micro-invasive glaucoma surgeries that allow for greater safety profile for our patients, I think we're moving towards a more personalized approach.
Yeah, Erin, how about you in cell and gene therapy? What have been the recent advances that we can talk about from an innovation standpoint?
Yeah, I think one of the biggest sort of milestones to me in the cell and gene space was the approval of Luxturna back in 2017. Obviously this is an AAV-based treatment for patients with RPE65 mutations in their inherited retinal disease, but not necessarily transformative from a sheer number of patients, but I think it really was the proof of principle that you can take a novel, innovative modality and bring it to market. And I think that really stimulated a lot of interest and in the field in order to use cell and gene. And so to me, that was one of the most influential.
Significant innovations.
Yeah.
Dr. Dana?
Most of anterior segment advancements really have been around microsurgery and so forth. The move away from whole corneal transplant to endothelial keratoplasty, where we do a transplant only of one layer. And then I would say also the really explosion of biologics in ophthalmology, including their application to the cornea. Now we have the first topical ophthalmic biologic recombinant protein growth factor, recombinant nerve growth factor, which we were involved in its development. So that's really been really important in cornea.
Yep. Peter?
Yeah, I think it's been very amazing 2 decades. It's hard to follow the literature. In the field, I think that I'm in for the cell therapy, one of the most exciting innovations is the work in limbo stem cell transplantation. Just the very fact that you can take the cells, expand them ex vivo, and put them back into the patients, it's amazing because as with gene therapy, as with Luxturna, this therapy never intended to target large cohort of patients, but it got us thinking about regenerative medicine, regenerative ophthalmology approach in our field.
Yeah. So let's focus a little bit on the front of the eye work, and obviously, Dr. Dana, in your realm, corneal work has changed so much. Corneal transplantation, keratoparesthesis, regenerative approaches. These are going to address many of our global blindness issues. But there's barriers to a lot of this stuff. It may be scientific, it may be regulatory. What sort of, kind of barriers do you kind of foresee in the near-term future, and is there a way out of them?
Thank you so much, Rishi. Very important question. We've got more than 20 million people blind in one or both eyes from corneal disease. We know there are 12 million who need corneal transplants, and we can only do transplants for 200,000 people now, so only 1 out of 70 people. So obviously, cost is like a major issue in terms of delivery of healthcare, including surgical care. In regards to new therapies, the principal barrier really is not science. We've broken many of those barriers. We know how to, as Petr mentioned, we know how to grow, cultivate, and expand allogeneic or autologous stem cells. We know how to grow autologous or allogeneic or iPSC-derived RPE cells. But these costs are really incredibly prohibitive. It's become very difficult, especially in the current environment where we have startups with amazing IP and other assets, but they're having a really hard time raising funds to develop their technologies. I think that's the biggest impediment right now for advancement. The microeconomic situation is difficult right now with the higher interest rates. And therefore, a lot of the investors are relatively, I would say, conservative. They don't want to get into technologies that are further out than 1 year from IND, and that really creates important barriers.
Mm-hmm. And what about scalability? When you look at sort of the ability for us to, develop, these treatment options, is there a lack or issue with scalability in these models?
Yeah, scalability is really, really critical, but I think whether, again, for example, in cell-based therapies, whether they're iPSC-derived or allogeneic-derived, we know how to develop technologies where we can have off-the-shelf therapies for our patients. It's not quite as scalable as developing a new pharma where you just, develop massive amounts of a small molecule, but we know how to do it. But again, the cost of going through that whole development, regulatory process, and then CMC, and then maintenance of inventory is, again, very challenging.
Yeah, that's great. Well, good answers too. And certainly feel free to ask questions, and we're going to take them in the live Q&A session as well during our session today. So let's focus a little bit on Dr. Kimbrell, your work. And you mentioned Luxturna, how much of an advancement it had been because of the first ability to have gene therapy come to light in our practices. But there's lessons learned in translating these therapies. Can you talk a little bit about what you're doing, especially in your role at Astellas, that is helping to understand better these sort of translational issues?
Yeah, thanks. I think, because in cell and gene there's really a lack of standardization in the field, I think one of the lessons learned would be when you have, a change in your process, when you're looking at something for the first time, it's really important to go early and often to talk with regulators as they recommend. And I think the other thing that's sort of an Achilles heel, because in cell and gene, some of the processes are still quite manual, is that you want to make sure that your critical quality attributes and your critical process parameters are linked, right? And I think that this comes to be incredibly important when you're talking about scaling, because even just the slightest change in your process may have an impact in your CQAs. And so you want to make sure that your processes are really robust so that you don't run into some manufacturing issues as you're trying to scale up to move forward.
And we've been focusing on gene delivery, but we haven't spoken about, gene editing and sort of those regenerative processes. Are those different in your mind as far as what those might be?
I think so. I think, for gene editing, certainly there's a lot of things that need to be considered. I think for one, for example, you could think about gene editing and the ability to use those editors to make precise genetic modifications for a small number of patients, but that becomes very challenging in order to fund that work, right? Other companies, certain companies like Sparing Vision, are trying to take more of a gene agnostic approach to widen the number of patients that may be amenable to their therapies with their work with rod-derived cone viability factor, which they're using as sort of a cone salvage approach. But in addition, I think we're also using gene editing at Astellas to help with our cell-based therapies and the creation of hypoimmune cells so that we can use allogeneic cells in a larger number of patients by opening up the possibility of having those cells not be subjected to immune rejection. So I think that there's so many different ways to use gene editing that are different than your standard AAV-based therapies. So a lot of important considerations.
Yeah, great commentary. Nazli, well, let's focus a little bit on glaucoma. Obviously, it's been one of the diseases we've had a tough time treating. We've obviously developed a lot of interventions. Some of them are not so perfect now, but we also know a lot from the artificial intelligence work that's happening, big data work that's been happening now and helping us better understand the disease insights as far as this. Can you give us some of sort of those takeaways maybe you might have learned in the past few years on AI and big data to apply to glaucoma that we've learned a lot about how we might address the problem?
Yeah, so I think, with the advent of large data sets now with, genomics data and large imaging data sets that are linked and electronic health records, we can better understand. So the problem with glaucoma is it's a complex disease. And in complex diseases, the difficulty that we have is binning all the patients into a single category, when in reality, it's a collection of different phenotypes and different diseases. And that's what we're trying to understand more with the help of these large imaging and genomics data sets, is what are the different phenotypes and how can we understand disease pathology better?
Great. And are we there at a point now where you feel like we can potentially personalize therapy? Do we know that certain patients are going to progress? I think that there's been a really interesting paper that came out this year in the glaucoma research that talked about, our patients not as progressing maybe as rapidly as we thought, and so is this testing worth it to us to look at? But we know, obviously, when we see patients in clinic, they all seem to progress very rapidly, and that's a big concern. It's a very controversial paper that came out this year, and I'd love for you to comment on sort of like, are we there at a point where personalized medicine really can be brought to clinic, or what are the sort of key factors we still are missing maybe from some of the points of that?
So I would say yes and no. I think one of the things that has really made a huge impact and is close to being clinically implemented are things like polygenic risk scores. We do know that we can risk stratify patients based on their background genetic risk. We can also understand sort of ahead of time what are the visual field or OCT phenotypes that may predispose someone to faster progression. So I say we've made a lot of strides in that direction. There is still a lot of gap because there is a limited amount of linked data sets that we can work with, and I think that's really where the limitation is in the research at the moment.
Yeah. Let's switch gears a little bit to Dr. Baranov. Retinal regeneration, pretty much the holy grail of what we hope to achieve. They had an audacious goal during the Biden administration of the ocular transplant. We were nearly not close to that at all, but maybe we're closer to retinal regeneration or retinal transplantation or replacement. What are your thoughts on sort of where that field is going? Is the biology helping you? Are there aspects of it that are still major challenges you're facing in that sort of realm of things?
Yeah, I think that one of the reasons why it's holy grail and still has not been achieved yet is that the retina does not regenerate, right? So we're born with 100 million cells, we lose them every year. Once they're gone, they're gone. So that's why I think the strategy is through cell replacement or ocular transplantation. ARPA-H has a program, NIH, National Eye Institute, they have Audacious Goal initiative to support this work. Companies like Astellas, Sumitomo, Blue Rock, they are invested in running clinical trials with stem cell-derived RPE and neurons, and I think it's achievable within 5 to 10 years.
Mm-hmm. So there's aspects of this, right? There's obviously the cell survival, there's the integration, and then there's the connection, right? So
Mm-hmm.
it's just not that we put the cells in, they have to actually talk to each other. Which of these three is solvable, and which one is just the most difficult from your research work and kind of what you've seen in the field, maybe?
I think it's like all or none. So we either solve them all, or like solving one won't help us.
Right.
And as you said, with some of the, depending on the cell type you're using, if you're talking about retinal pigment epithelium, then you need them to connect with each other to form these tight junctions, and to structure as a tissue. If you talk about photoreceptors, they have to connect with a cell within few microns, and that's why photoreceptors are in clinical trials right now, When you talk about ganglion cells, so these are the large neurons lost in glaucoma, they have to grow 10 centimeter long axon all the way to the brain in the environment that is not very friendly. So we still have to solve it, and we have to solve all the problems, because if we can make the cell survive, but if it doesn't connect, then we don't need this cell in the eye.
Yeah, yeah. And talk about how maybe AI has helped transform, or has it not come to your field yet, or maybe it has, and tell us how it is helping you kind of with the next future of what you're working on and doing.
I think we were talking about that before the session. Now it's AI, but it used to be data science or machine learning or convolutional neural networks, or you name it. So I think it helps us a lot, especially to think about scalability and how to make it affordable for patients, because when you try to envision autologous-derived therapy, when you take a blood cell from a patient, convert it to pluripotent stem cell, then differentiate into the retina, then put back into the eye, you need either image-based analysis or some single-cell-based analysis that would be performed by the AI, because we just don't have enough workforce to do it for every single patient.
Dr. Dana mentioned in your discussion about the environment we're in right now, obviously with cost of drug development being significant and trying to find investors in a milieu or market where it may not there be that aspect of interest in putting money forward for these sort of things. Let's talk a little bit about, as a panel, maybe how we're approaching some of those financing options. Why are companies investing in this? What do they need to see maybe to invest in some of these programs? Maybe Dr. Dana, you can start, and we can also hear some of the panelists about that.
Thanks, Rishi. That's a really complex question. I think the model that we saw from like 2005 to 2020, where larger biopharma and biotech concerns, like the Amgens and the Biogens, were essentially largely pulling away from doing fundamental science and then passing it on to smaller companies, it was really working. And what we've seen, especially since 2021 and beyond, when the markets reached a peak and then they went down and interest rates started to go up, is that there has been a major, I think, pullback from funding higher risk technologies. Obviously, higher risk usually means, if they're successful, higher reward at some point. But when that reward comes 6, 7, 8, 9 years later, with interest rates of 6, 7%, that's going to become very difficult, because you do the NPV calculations, it doesn't really work too well. So I think what we need to work toward, hopefully, is a combination of things. For academic medical centers, I think we need to remain engaged with clinician scientists who understand both science as well as the clinical indications that these technologies are supposedly addressing, and when there's a misalignment, that leads to problems. Second, I think it's really important, those of us who work in institutions like MGB, Mass Eye and Ear, where there is sometimes resources to go toward very potentially productive and promising technologies, so internal support for assets can really be helpful in getting them from preclinical POC to maybe IND enabling studies. That's going to be very important. And then, hopefully we just have to weather, through this current environment and hopefully wait for better years to come.
Yeah. Dr. Kimbrell, you're making obviously decisions with the company to say, Stargardt's disease is probably 5 or 6 different potential phase 2, phase 3 trials. What would be an interest of yours in your field, your company's field, to say we're going to be the next in Stargardt's disease when everyone else is also going after potentially?
Yeah, I think it's an interesting question, and one that we talk about a lot. What is the value proposition? What's the longevity or the durability of the therapy that you're hoping to put in or to deliver to these patients? They're younger than those that we may be treating in geographic atrophy, and so will you have that durability of response over a patient's lifetime? Will you be able to redose if needed later in life? So those are some of the considerations. But I think in more general terms, as we think about the potential and the promise given to cell and gene therapies, in order to be able to bring this to market, I think we really need to have those put the pieces together between the patients, the physicians, and the payers to really make sure that we have alignment on what can these therapies bring to the table, and how are we going to get a reimbursement model that will enable better access and enable them to be paid for.
Maybe I could ask you a follow-up question, came from the audience member, it kind of relates to this, is obviously we talk about delivery of genetic medicines, but is there an ability to avoid viral vectors? I think the concern has always been they go in, but you don't have an ability to turn them off potentially. Can you talk a little bit about that?
Sure, I think there's a lot of ongoing work to use non-virus-based delivery methods, right? Lipid nanoparticles, extracellular vesicles, polymer-based nanoparticles, cell-penetrating peptides. There's lots of interesting technology in the non-viral space right now, and I think we need to consider holistically what are the best approaches, and certainly the AAV space in particular has had a lot of negative publicity lately, and so maybe that will be a push to consider these alternatives to virus.
Yeah. Nestle, you span obviously the individuals in the research realm, the clinical realm, obviously work with some of the industry people and working what you're doing. Can you give us a sense of sort of like the work that's going on? How do you enable that cross collaboration? And I'm going to ask a follow-up that the audience member asked as well, but love to hear kind of how you're managing to bridge all those gaps in the world.
Yeah, so I think in my mind, maybe talking mostly from my own experience, I think it's critical to have interdisciplinary education in academic centers, right? Mm-hmm. Having clinicians that can speak the language of data science, that can speak the language of public health or genomics or other factors that allows us to ask those clinically important questions, but also bring it into, a translational reality. So I think that's a big thing. And then having actual formal mechanisms to foster these collaborations. So centers, there is, for example, the Collaborative Ocular Imaging Association, where we bring, people from different fields together. We can think through these problems.
And then a follow-up would be about, sort of the imaging you're doing, glaucoma biomarkers. What have you been able to find and in your work? And also, is that helping us kind of better define those progression patients potentially?
So what we have worked on up to this point is understanding different image-based phenotypes for our glaucoma patients, and we are using that for genome-wide discovery. So we're able to see that, using more specific phenotypes than just yes glaucoma, no glaucoma is able to allow us to discover more pathways, more genes, and more risk variants. And then, ultimately we want to move that towards understanding the genetics of glaucoma progression, which, I feel like is sort of the holy grail, right? If you can tell ahead of time who's going to get worse, then we don't need to do all the unnecessary testing.
Right. Petr, when we talk about, and any of the panels can talk about this, is in gene therapy or regenerative medicine, there's obviously innovation and safety. How do we find or balance that in your mind? What are the things you're looking for from a safety perspective that help you better in your level or even the clinical level to understand that better?
I think there are going to be a lot of failures. So in terms of safety, I would say we shouldn't be afraid of a failure, but we have to be very transparent about the data that we collect, so we can go back and I can share this data. And Erin mentioned standards, especially in cell therapy. These standards are still being developed. So being able to go back and assess what went wrong and why is one good thing. And the second, within the eye, it's an approachable, very approachable part of the brain. So I think that's where imaging plays in our favor, that we have laser, we have all the imaging tools we need to continuously monitor neuroprotection or biologics.
Any of our panelists want to talk about safety in their patient populations with some of these gene and cell therapies?
Go ahead. Well, I mean, it's again, a very complex situation, and we can look at it multiple ways. I think, number one, when we think of degenerative conditions, we need to recall that these patients, these hosts, had normal cells, but we can't only just replace the cells, right? We have to also think about the environment in which we're putting these cells, which are prone to also dysregulation. We also can potentially minimize the risk, if you will, by doing modulation of the immune response by creation of hypoimmune cells, or we can use progenitor cells that are partially differentiated as opposed to like less controlled cells that sometimes emanate from iPSC-derived cells without adequate safety measures. And then I think lastly, in regenerative medicine, there are approaches that are not cell or gene therapy based. There are specific factors depending on context that we know mediate, for example, the pro-homeostatic functions of MSCs, right? So if you know what it is, then you can develop a biologic therapy which is molecular based as opposed to relying on the whole cell and the complexities around the cell.
I was just going to add, I think it's, from a safety perspective, it's important to understand your patient population. And, they're, depending on what indication you're in, they may be less tolerant of certain types of therapies. And so you need to understand what those risks are for your patient population. I think another critical thing, at least in the AAV space, is as the field is learning and progressing, being able to have engineering to help better targeting for the tissues that you want to reduce those off-target risks. And then lastly, I think for both cell and gene therapy, it's the quality of the manufacturing and making sure you have really sensitive assays built around safety aspects of the assets that you're developing.
Yeah. This is an audience question again, and I think it's something we can all kind of comment on. I'm happy to comment on this myself. Recently approved therapies for complement inhibition were approved, and they were approved on structural biomarkers and not vision necessarily. It's probably the first time we've had in our field sort of that approval process. And is that biomarker sufficient indication for approval without functional benefit? And we've dealt with this in the corneal realm, obviously. In the retina realm, to my extent, I mean, I can say that actually the vision may be the poorest biomarker we have for geographic atrophy, to be quite honest with you, and maybe that wasn't the best functional marker to determine. I think we're learning more about it from some of these complement programs about what the biomarkers should be, and certainly it's probably low luminance visual acuity, which has been correlated to some extent with some of these complement therapies, but there's other, better visual methods that we have to develop for these conditions that really have to be out there. But I'd love to hear kind of some of the clinicians' thoughts on these structural approvals, but not necessarily functional benefits per se.
Yeah, I mean, personally, I see this transition that has occurred at the FDA to be a positive one.
Yeah.
So I happened to be involved in arguing when we went to the FDA for the recombinant therapy for corneal defects. The FDA initially said it has to be vision. And then we said, well, if you have an epithelial defect that is not healing in the central cornea, obviously that can't be good for vision. So if you just rely on vision, you might be like really missing important changes. So I think similarly, the adoption in some cases of microanatomical changes for the retina, especially like in the GA space, really has been very impactful, I think, and a positive one.
Yeah, I would add that, from a glaucoma perspective, especially when it comes to things like neuroprotection and neuroregeneration, relying on visual field metrics might be a little bit more difficult, but of course much more important than OCT-based metrics, for example, or imaging-based metrics.
Yeah. And talk to me about what's changed with the regulatory side. Maybe both of you can comment on the length of studies. That's changed. I know in retinal studies, they used to be 3 years long, very, very arduous process, expensive process. That's now been obviously transitioned to sometimes a year or even 2 years with safety data, a year with efficacy and 2 years with safety. Comment a little bit about what's changed in your fields in cornea and glaucoma in regards to those FDA endpoints, maybe.
It depends on the indication. So The general rule for topical corneal indication is to have a minimum of 1 year safety study. So typically, you run 2 pivotal trials for efficacy. I mean, these could be 8 weeks, 12 weeks, 16 weeks, or longer. And then you have to have a minimum number of subjects go through 1 year of therapy to ensure safety for chronic administration of drug.
And do you think that's going to change or stay the same, or what's your thought process?
Well, it's been working, and it's been working without too many like black labels coming up later, so I think it'll stay.
Yeah. Natalie?
So from a glaucoma perspective, obviously glaucoma can be a very slowly progressive disease, so this becomes an issue, right? How long do you need to study a patient to understand the effects of neuroprotection or gene therapy or any other therapies that are coming into the market? And I think even though the regulations have not quite kept up with that, I think one way that a lot of companies are addressing this is by enriching their samples for people that are going to get worse faster. And we can do that by understanding biomarkers from baseline images or from patients' genomics to understand risk profiles and preferentially look at those patients that are more likely to have change.
Yeah. Petr, how about in your work with cell regeneration and cell therapies? What are sort of the approval processes you see, and do you think they're going to be long or the short, or how is that going to look like for your perspective?
Yeah, I would say approval process develop as we go. So I was fortunate to be involved in cell therapy development for retinitis pigmentosa that went to a clinical trial. And it was very interesting interaction with FDA because we would give them data and they would ask more, and then we would change the requirements for the submission as we educated them on the material and on the disease. So I think it's going to change. I think we're going to see more individualized biomarkers from the work that Nestle is doing, that we can select a cohort of patients and focus on cells on individual level rather than on a whole tissue level.
Yeah. So maybe we'll just go a last kind of commentary on the panel because we're going to end this session. And thank you all for your attention. Thank you for the questions. So start off with Dr. Zebredas and ask you about sort of the holy grail for your therapeutic area. What are you looking for the next generation of therapies that would really change your patient and clinical outcomes?
I think as a clinician that spends my time either treating patients who are rapidly losing vision from glaucoma versus those that never progress, I think the holy grail would be able to tell or risk stratify patients and to be able to offer more personalized treatment approaches. So what I see all of this data analytics and multimodal models creating is essentially a clinical decision support system, right?
Yeah.
So you can say to a patient, this is your % chance of progressing over the next 2 years. This is the next best recommended therapy, and allowing the physician to then make that decision, but also using all the available data to better serve our patients and ultimately save vision.
Yeah. Erin, how about in your realm? What is the kind of thing you hope to achieve in the next 3 to 5 years with therapeutics?
Yeah, thanks. I think for us, we're hoping to get to clinical proof of concept with our pluripotent stem cell-based therapy, right? It's been a long time coming. It's one step forward, one step back, but I think we're making good progress now, and I'm excited for the next 3 to 5 years because I think we're gonna be able to achieve that milestone and see where we are.
Okay. Reza?
I think biologically, our goal is to reestablish homeostasis in the cornea, as well as in the retina, as well as all tissues, of course. And we've been really focusing on the crosstalk between the immune system and the neurons, and especially the afferent nerves that innervate the cornea. There's a significant crosstalk, and so we've been able to identify factors that can basically suppress immunity by promoting immune regulation, also providing support for neuronal regeneration. And then that improvement in that crosstalk will lead to regeneration of epithelial cells and endothelial cells, we found.
Yep. Petr?
I would go a bit technical and say that I'm looking forward to a switch to in-process quality controls for cell therapy. So right now, for every drug product, we have to collect it at the end of the manufacture process and do a flow cytometry, PCR, your favorite method of analysis. I think we already possess the technology to do it as we grow the cells. So then the patient does need to wait for the results of the end of stage quality control. And also we can scale it up and make it accessible.
Yeah. Well, I want to thank my panelists for this wonderful discussion. Thank you for the audience for coming and participating in it. For the sake of the next panel, we will end the session. Thank you again for watching.